Hi, Mike Dolinski here with Taurus.ag. Just want to take 10 or 15 minutes to talk about how cereal plants complete their life cycle, filling the grain before they actually senesce and die, so that we can harvest that grain. And I want to look at it from the plant's perspective, so you can understand whether something like grain fill, a product that we sell, may fit your operation. Now, I'm going to start out with this because, I'll be referring to it throughout the presentation. What we need to do is keep the plant alive as long as we possibly can so it can remobilize the nutrients that it has accumulated over the summer and move those nutrients into the seed to complete filling. And we know that nitrogen and, and basically potassium are the two major nutrients after carbon, hydrogen, and oxygen that are required in plant cells. So by providing those late in the season, generally at fungicide time, if you're spraying for fusarium head blight, just at early flowering, for example, or mid flowering, depending on your approach to managing your fusarium, are key to sustaining those leaves and keeping them green so the plant doesn't start breaking them down and removing those nutrients going into grain filling. So you want to keep those nutrients there. Eventually they'll be all pulled into the seed. But along with that, besides just feeding the plant, we want to deal with stress. The plants are always in stress, and sulfur and potassium are really key in stress management, along with a bunch of amino acids and even vitamins like ascorbic acid and the vitamin B complex. So those are included in this mix and go on later on in the growing season. Now let's take a look at what actually happens with the plant. Now, what does it take to increase yield? We know that it takes spikes, more seeds, which is too late now if you're going into flowering. And, that's going to vary across the prairies this year with the moisture and temperature and water conditions. But we got the potential for a big crop because we have some moisture in the soil now. So when you're going into flowering, uh, head formation and so on, you've already got all the seeds there. So now all you can do is help plants make bigger seeds. That's where you're at. Up until flowering, you have been supporting through your nutrient package and what the plant can absorb from the soil that you haven't applied, but through your nutrient applications and your protection of your plants, have been developing the reproductive parts, the leaves, the stems, and the roots. And because the roots can feed themselves, the leaves have been really key in photosynthesizing, providing those sugars to the roots and through the rest of the plant, and using sunlight to make photosynthate, and that's basically what you've been doing. So let's take a look at this specific time of grain filling. That's just after flowering takes place or pollination. So this explains it fairly simply. Grain filling is a stage of growth after fertilization that determines your grain size and density. That's all we can do at this point. During this period, the plant transports nutrients stored in its foliage to the head for grain development. Pretty simple, right? Now, this is all based on a source sink concept. Throughout the plant's growing life up until this point, the source of nutrients or photosynthase for the plant has been photosynthesis. That's the source in the leaves, in the stem. The sink have been the roots and the growing vegetative part. When we hit pollination, hormonally the plant changes, and now the focus sink is the seed. Now the plant is photosynthesizing and it's making a decision, do I support roots or do I support my seed? Well, it's going to support seed because that's the objective of the plant, is to produce seed at the end of the day. So it's shut off some of the supply of photosynthase to the roots to focus on filling the seed. Let's take a look at how this happens. Now, a lot of people think that once the head emerges from the basically the boot of the flag leaf, it pollinates. That's not what happens. what happens is the plant pushes what is called the peduncle, this last internode between the head and the last node on the plant. There's only 3 nodes on a wheat plant generally, and that last one's the long one that pushes the head up. So you can see here that when the head is formed, it's slowly pushed up by expansion of this peduncle, and it'll move it up about 10, 12 inches above the flag leaf. And that exposed peduncle is photosynthetic, has a lot of stomata, and it's very active. Here's a closer look at that. You can see the flag leaf and the peduncle supply most of the assimilates for the filling of grain right up until maturity and basically cell death, because the plant is programmed to die just like all sort of living things. It has a lifespan. Our objective to help fill that grain is to prolong that death as long as possible so that the plant can move those nutrients up and photosynthesize as long as possible. And the plant will scavenge all those nutrients by killing the cells in the lower leaves and moving those nutrients into the stem and moving them up into the seed head. And the last part to die on the stem is this little part right here just below the head, because the plant has to use the stem right till the end to move all of those nutrients up into the head. So let's take a look at how this happens. Here's the head, and this group of seeds is called a spikelet, and each one of them is called a floret. And this spikelet sits on a node like this running down the middle of the head. And when we look at the number of seeds produced, we can see that pollination starts in the middle of the head, and here's where we're at. We get 4 seeds, 4 seeds, 4 seeds, and 3 threes, and 4 here, moving up and down until at the end, the plant is running out of nutrients, and it's basically programmed this way. We get fewer seeds. And in the tiller, we get a lot fewer seeds because it comes on later, and the primary head will pull the bulk of the nutrients from the plant as it's maturing. So let's look inside that floret, and we can see here we've got the anthers here, the stigma, and this is a little tip for you guys that are going to spray for fusarium head blight in terms of timing, because the plant will pollinate, and I'll just show you how that happens. But when you're looking inside here, you'll see that the anthers, when they're not mature, are green, and as they mature, they turn yellow. So I'll show you that a little later. But if you look inside, you can see that the pollen is already on here, on the stigma, and it'll move the pollen tube down here and fertilize the embryo down here. Now here's your tip. When you see this first anther out, that tells you, and it'll be nice and yellow because it's still fairly fresh, and then as it ages, it'll turn white. So you can tell basically, when it was pollinated. And if you look inside, you'll see what I just showed you, those anthers and remaining pollen, anthers that haven't been ejected yet. So when you see that, which is usually at about the time that the elongation, the peduncle, is done, which takes about 5 to 6 days depending on the temperature, you're going to be spraying pretty darn quickly to control your fusarium. So let's take a look at how that seed develops after pollination, because this is really the key thing. You want to get it fully filled like this. So right after pollination, the plant accumulates nothing but proteins and the necessary ingredients to make the seed. You can see there's really no development of the seed until it gets a little later on, and it's nice and green, and then eventually it fills up, starts browning, and gets mature. So what we have to do when we're talking about a source and a sink, we have to realize that the seed is full of endosperm, and it's mixed with proteins, and those are storage proteins, gliadins and those kinds of things. They're different than the proteins used to run the plant. And the starch is stored as either amylose or amylopectins. And you can see here that just like chloroplasts are used in photosynthesis, there are amyloplasts that are formed to store those storage endosperm. I was just mentioned amylose and amylopectins. So you have to build those. That's what happens in the early stages of development after the plant has started to form the seed. If you don't have a lot of amyloplasts, you are restricting your sink. So you can either have seed size and productivity in your grains restricted by source or by sink. If you haven't got enough source material to fill the sink, you get a smaller seed. If you don't have a big sink, you get a smaller seed. It's just that simple. You can see here that this seed is developing. This anther was trapped in there. It never got ejected yet, but you can see it's yellow. So when you look into that floret and the seed, the anthers are yellow, you know you're close to pollination. This is a little later. And if you take a look at the actual seed that is developing, this is from the same head, but this is a seed developing in the middle of the head, and this is a seed developing at the bottom of the head. You can see that it's quite advanced in the middle of the head sooner. So when you look at that spikelet, you can see that this seed and this seed, these two first seeds sitting on that node, get the most nutrients and will be the biggest and almost always the same, followed by the third and the fourth. The fourth will be the smallest. This will be a little bit smaller. Now, generally, you're going to get 4 seeds in the middle of the spike. You rarely get 5. If you get 5, you got a bumper crop coming. If you get 4, that's a sort of an average or a good crop. If you get 3, that's a poor crop. It's just about that simple. And the more 4s you get, the more seed you're going to have, and basically grain filling is about filling those. So you can see that now the seed here is starting to form some structure. You can still see the remnants of the anthers or the stigma. Sorry, I get those two mixed up a lot. And as it's developing and starting to fill the endosperm here with all that starch and mixed in with the storage proteins, we get the germ that then develops. So the plant has this reproductive component developing there as we start building that seed. And you can see here, nicely filled. The entire capacity is loaded with granules, and there you have your seed filled. Nicely filled. You want it fully plump, and then eventually it dries down and you're ready to harvest. And you can see these big seeds here in the third seed. And what I tell farmers if they're going to use their own seed is always go after these 2 seeds, clean your grain really hard, go for the big seeds, and you can use those as your seed source. Generally, we want people to use pedigree seed, but a lot of folks across, the prairies use their own seed for a couple of years until they change varieties. And you're basically done. And this table just shows you that the plant mobilizes darn near everything, especially wheat, all the N, the K, S, the phosphorus, the magnesium, the calcium. Strangely, it moves calcium. Most people will tell you it doesn't move calcium, but it doesn't necessarily get the calcium out of the fully out of the structure of the leaf. It gets it from the cytoplasm. The soluble calcium that is run there. Calcium is really important as a signaling agent, so there's always a well-controlled level of calcium in the plants, and it even moves micronutrients late in the growing season. Otherwise, we would get them stuck there. So that's really important. And then we have harvest going on, and we're done. So here we are back to the beginning. We have these nutrients that we can apply to sustain the flag leaf and that peduncle. The peduncle will produce photosynthesis long after the flag leaf has been exhausted and broken down. That peduncle is also very close to the seed, so it can move it up more efficiently late in the growing season. We need that potassium there to manage the stomata. And that peduncle is loaded with stomata just as all the other plant cells in the leaves and so on. All the tissue has got those stomata that regulates the movement of nutrients and water. These amino acids, iron, are involved in stress management. The nitrogen and sulfur are used to rebuild proteins there. We know that needed. And if we don't keep the nitrogen levels up late into the growing season, the plant will start scavenging the leaves and breaking down early, reducing the length of time you have to produce those photosynthates to fill those amyloplasts and make a big seed. Remember, the plant knows what to do. We're just going to help it. Hope this helps you understand how that filling takes place and how we can handle that late stage in growth and add to our yields. Thanks a lot.